Blood is the largest organ in our body. 10 pints of life force coursing through thousands of miles of arteries, veins, and capillaries. Those blood vessels carry blood to every other organ, keeping them functioning and thriving. Our red blood cells carry oxygen and nutrients. Our platelets stop bleeding and help to heal our wounds. Our white cells protect us against infection. And the yellow liquid plasma in which they are suspended carries an array of proteins that regulate bleeding and clotting.
The white blood cells are an important part of our body's immune system. Neutrophils are a special group of white blood cells that play an extremely important role in protecting the body against infections. Mature neutrophils do not divide. New neutrophils are constantly formed in the bone marrow where they develop and mature. Mature neutrophils are released into the blood where they circulate for 3 to 12 hours and then move to other tissues where they survive only 2 to 3 days. These mature neutrophils can be found in large and small blood vessels, as well as in the tiny capillaries in different tissues. In the bloodstream, neutrophils act as surveillance cells searching for infections. Neutrophils can sense a site of infection because of chemicals that are released by the bacteria. Upon detecting these chemicals, neutrophils slow down and begin to stick to the walls of the blood vessel. They then squeeze between the cells of the blood vessel wall and enter the infected tissue. Here, the neutrophils crawl towards the infection as they continue following the chemical signals. At the site of infection, bacteria are coated with antibodies of the immune system and marked for ingestion. The neutrophil recognizes the marked bacterium, engulfs it into its body, and traps the bacterium in a sac called a phagosome. The neutrophil contains pouches called lysosomes, which are full of digestive enzymes and chemicals that can kill bacteria. The pouches, or lysosomes, then combine with the phagosomes containing the bacteria and release the enzymes and chemicals, resulting in the ultimate death of the bacterium inside the neutrophil. After killing and digesting the bacteria, the neutrophil is spent. Having completed its duty, the neutrophil shrinks in size and breaks up into smaller pieces that can be recognized and eaten by other cells.
Membrane factor. This factor mediates the linking of platelets to collagen via a specific receptor in the platelet membrane. The resulting change of shape of the platelet from its resting state into the dendritic form indicates activation. The activated platelet in turn releases prothrombotic molecules such as adenosine diphosphate, ADP. By binding to its receptors, ADP induces aggregation. And recruits further platelets to the site. Thromboxane is another important mediator of platelet activation and aggregation. Under its influence, the platelets cross-link with each other. These interlocking mechanisms cause platelet activation to snowball. The clot grows rapidly. Activated platelets also trigger the coagulation cascade and thus the formation of thrombin. Thrombin, in turn, stimulates platelet activation even further, a continuous feedback loop. Additionally, thrombin induces the formation of fibrin for the mesh stabilizing the clot. The self-reinforcing process of platelet activation, crucial in the formation of blood clots, is an obvious therapeutic target in conditions caused by inappropriately triggered blood coagulation.